All right, I think we can get started with a few announcements before today's lecture in the Cognitive Ontology seminar at the Center for Philosophy of Science online. So before introducing our speakers before Adina introduces our speakers, I wanted to tell you a few things about the events at the center this week and the next Cognitive Ontology seminar meeting. On Tuesday, Mac Pedroso, a center fellow, will be giving a lecture at noon. And on Friday, Diane O'Leary will also be giving a lecture at noon, also online. If you are interested in their lectures, please go to the center's website, to the calendar, where you can find the title, the abstract, and a link for registration for the Zoom event. The next meeting of the Cognitive Ontology seminar will be next week at 1:30 again, and we'll have two lectures, one by Vincent Bergeron on carving the mind at its homologous joints, and one by Javier Gomez-Lavin on productive pessimism and new ontologies of cognition. And again, from 1:30 to 3 on Monday next week. If you're interested, again, please go to the center's website and you'll find, go to the calendar on the center's website, and then you can click on a link that will bring you to the abstracts as well as to a link for the registration to Zoom events. And without further ado, I'll let Adina Roskies, my co-organizer, to introduce our speakers today. Adina.
Great. So welcome, everybody. I'm really pleased to introduce our speakers for the next keynote lecture of the series. They're going to give a joint presentation. One speaker is Michael Anderson. He is a Canada Research Chair and Professor and Chair in Philosophy of Science at the Rotman Institute of Philosophy, which is part of, I think, University of Western Ontario, which may be renamed Western University. Is that right? And Mike's, I think, familiar to almost all of you. He got a PhD from Yale University and works at the intersection of psychology, neuroscience, computer science, and philosophy of cognitive science. And probably many of you are familiar with his most recent book, which is called After Phrenology, Neural Reuse and the Interactive Brain. Our other speaker is Paul Cisek, and he's an associate professor at the Department of Neuroscience in Université de Montréal, which Edouard could have pronounced much better than I just did. He works in neural, behavioral, and computational studies of decision-making, planning, and movement control, using a variety of experimental techniques as well as computational modeling. And together they are going to give us a talk, 2 approaches to reforming the taxonomy of cognitive neuroscience. So I'm really pleased to have both of them here, and I'll turn it over to them now.
Thanks so much, Adina and Edouard. When I accepted the invitation to give this talk, or a talk, in person at Pittsburgh, one of the things I was most excited about was actually bringing Paul Cisek's work to your attention. Now, COVID took that possibility away to be there in person, but it does give us the opportunity to let Paul share his work with you himself. So I'm quite happy about that. So without further ado, let me get going here. So as Adina said, we're going to talk about 2 different approaches to reforming the taxonomy of cognitive neuroscience. Here's outline of the talk. I'm just going to talk briefly about why we're bothering to think about reforming the taxonomy of psychology of cognitive neuroscience, and then the promised 2 approaches. So I will briefly go over the typical approach that people have taken to this reform in the cognitive neuroscience, and then turn it over to Paul for his, I think, really interesting and quite new approach. And then we'll talk a bit about how we might bring these 2 different approaches together. So capturing the motivations. And I'm going to go through this quite quickly. You know, obviously people tuning into this seminar are already, I think, fairly aware of the issues. At least I'm hoping that's the case. So why are we bothering to think about reforming the taxonomy of psychology as it relates to our understanding of the brain? Well, one of the initial and ongoing motivations is that the expected one-to-one mappings between cognitive operations and neural regions has just not come to pass. According to currently available data, the mapping is in fact many to many. It takes many different regions to implement any particular cognitive operation, and each neural region seems to be implicated in many different cognitive operations. That is to say, individual regions of the brain are used and reused across different task categories. And differential function is not the result of differential activation so much as the result of the dynamic reconfiguration of large-scale networks. Here's just one data point. I'm not going to go over this in much detail, but just as a way of illustrating what I'm talking about. So this is some work I did with colleagues a couple of years ago, where we took every voxel in the brain, that is every location in the brain. We looked at a couple of thousand neuroimaging experiments. We drew a 10 millimeter sphere around each voxel in the brain. We looked at the activations, the neural activations from the fMRI experiments that fell inside that sphere around the voxel. And we counted them and where they fell in this particular case, in 11 different cognitive domains. And that gives us the capacity to calculate the diversity of activations, the functional diversity of each of the regions of the brain. And so here's the histogram from that result. And you can see that, by the way, we scale from 0 to 1. So 0 would mean every activation we observe is in one category. So it's a functionally dedicated region. And 1 would mean it's basically equipotential. It's equally likely to see an activation in any of the categories we're looking at. So here's the histogram for that particular analysis. And you can see it's highly skewed towards the top end of the scale. That is, most regions of the brain are quite functionally diverse. So 0.76 is the median of this distribution. This is a voxel from left thalamus that has that value, just to give you some sense of what that looks like. And the way to read this spider plot is the further out along these axes you are, the more likely you are to observe activation in a particular category. In this particular case, not surprisingly for thalamus, we see a lot of activation, high likelihood of activation in somesthesis, that is bodily perception. But we also see activation in lots of different categories, including action tasks, including language tasks, and so on. Here's a very high diversity region. This is a voxel from left anterior insula. You can see that it's very likely to be activated in almost everything except, in this case, vision. And then here is a relative specialist. This is a voxel from left auditory cortex. And it's mostly dedicated towards to addition, although it has some stray activations in other categories. But as you can see from the histogram, almost nothing in the brain is actually like that. Most of the brain looks more like thalamus than like auditory cortex. So we have this this finding where, unlike the expectation where we would get these nice one-to-one mappings between cognitive operations and regions of the brain, instead we have functional diversity across the brain. Now, that could be because each part of the brain is functionally diverse. That's, of course, where I place my bets. But it could also be because we're querying the brain with the wrong set of basic categories. And if our categories cross-cut the ways in which the brain is responding to things, then you would expect indeed to see a diversity like this. It's just that the diversity would be an artifact of our categorization scheme and not something that's natively true of the brain. So this might give you a motivation for questioning the taxonomy we're using to study the brain. So that's one motivation. This is just, by the way, a different from a different study. These are functional fingerprints for left and right insula, the three main anatomical divisions of insula. The point here is just, again, that this is a finding that you can reproduce using lots of different methods, using lots of different granularity in the cognitive operations you're looking at, or the cognitive domains you're looking at. It's also to underscore that none of this is to claim that every part of the brain is like every other part. You can see these functional fingerprints differ from one another in considerable ways. Nevertheless, this finding of diversity is easily reproducible, even if it turns out to be an artifact of the categories we're using. And a similar phenomenon is apparent for psychiatric conditions. There is, in fact, no clear neural basis for most psychopathologies. That's been frustrating for biologically inclined neuropsychiatrists. It's to the point where the US National Institute of Health has made it a criteria for getting funding that you be looking for evidence for a neural system or circuit that plays a major role in the function that you are interested in studying. Here again, if you're not able to isolate a neural circuit that might be responsible for a particular pathology, that might give you reason to question the ways in which we're dividing up the pathologies. And then for the last motivation, and these are not to be, this is not an exhaustive list, this is just to sort of remind you of the kind of sources of motivation for this particular project. There's the computational theory of mind itself. Now, the computational theory of mind is the explicit basis for cognitive psychology. Cognitive psychology, which had its, started to take off in the 50s, was rooted explicitly in the notion that the mind, the brain, might be a particular kind of computer. And that in turn, cognitive psychology is in turn the explicit basis for cognitive neuroscience. Why is this a problem? Well, the computational theory of mind and cognitive psychology itself was formulated explicitly so as to be autonomous from neuroscience. The notion was that there was a level of analysis that one could pursue in a scientifically responsible way that could simply ignore the brain, which it treated as mere implementation details. Moreover, this is a reverse engineering approach. In other words, it's not an ecologically or evolutionarily grounded approach. It's a, let's throw stimuli at the brain, let's see how the brain responds, let's use that to figure out what the circuits are and look like. But it's something that is, again, initially was meant to be autonomous from neuroscience. Then we use neuroscience, but we use it as if we're just investigating a mechanism of a particular kind. And we're not taking into account the evolutionary history of that organ. And finally, if you spend time in the camps where I live, on the kind of radical side of the cognitive sciences, you might think the computational theory of mind is just false. And if it's false, it's certainly the wrong thing to be using to study the brain. So if you're a proponent of embodied cognition, or even sort of further to the left, ecological psychology, you think, look, computational theory of mind is just the wrong place to even be starting to do this. And the kind of analyze, decompose, functional decomposition that computational theory of mind advocates is simply the wrong place to begin the study of psychology, much less a psychology, psychologically grounded, neuroscientifically grounded psychology. And so that brings me to the empirical approach. Once you're motivated to change, to reinvestigate the taxonomy of psychology, how do we go about doing it? And I'm not going to go into much detail about this because I know you heard from Russ Poldrack not very long ago. And again, if you're already familiar with this debate, this is sort of just the standard way that people in this field have gone about trying to investigate alternate taxonomy for psychology that's more appropriate to neuroimaging or to neuroscience in general. So the current logic of cognitive neuroscience is something like the following, which is, given we know the labels assigned to cognitive processes by psychology, let's then use neuroimaging to find out the neural features or the neural activations or the neural mechanisms that characterize or allow those processes. What's happened now, though, is we're losing confidence in the accuracy of those labels. And once we lose confidence in the accuracy of those labels, what do we do? Well, one thing we can do, and this is work that's been done and is ongoing, is we can actually treat the brain itself as a task classifier. Suppose we don't know the labels. Nevertheless, the brain is differentially responding to the stimuli and the tasks that we throw at it. Well, then we can say, what does the brain treat as similar and what does the brain treat as different? And we can potentially use that as a guide to recharacterizing the taxonomy of psychology. That is, we can treat the processes like objects that we need to classify based on their neural features. So anyone who's even a little bit aware of developmental psychology will recognize critters like these. And the notion is that, in fact, they come in two kinds, glorps and dremps. And the question is, what's the difference between them? They've got lots of different features, colors, and shapes, and so on. Which of those features matters to their classification, and which of those features don't matter? So going from the multiple numbers of features that these critters have to the ones that matter to the classification, that's simply a process called dimensional reduction. And by the way, for those who are trying to figure it out, it's pointy feet versus flat feet. So a crash course for those in the audience who may not be super familiar with this way of proceeding. This is a crash course in vectors and functional spaces. So neural activity at a time can be represented as a series of numbers. So in fMRI, for instance, you can take every voxel, every location in the brain, assign it an activation value, and you get a very long string, maybe 20,000 numbers long, but a very long string of numbers. That long string of numbers is the vector that represents the activation state of the brain at that particular time. Now, that's 20,000 numbers. You can also think of that as 20,000 dimensions. That is a 20,000 dimensional space. Don't try to visualize it. But just think of, you have got 3 dimensions in normal space, and then you just add dimensions to that. So the spatial interpretation of a vector is a location in that high dimensional space. Now, we're going to do dimensional reduction. Big matrices of neural activation, right, this 20,000 dimensional space, you can often reduce those dimensions quite massively that allow you to capture the variance that you're actually seeing, the important variance that would tell us the difference between, say, neural glorps and neural DREMPs. So these big matrices, there are lots of different kinds of analytic techniques you can use, right? Reduction, classification techniques, factorization, similarity analysis. It doesn't really matter. Once you've got vectors, and once you've got vectors in a big functional space, there are lots of different mathematical techniques you can use to investigate the patterns of variance in those spaces. And that's indeed what people have done. So when we treat the brain as a task classifier, what's the first step? Well, we do this kind of dimensional reduction. What features actually define the brain's functional similarity space? What things are clustered together in one region of that space, and what things are clustered together in a different region of that space? And then why are they clustered in each of these different regions? What is it that they actually share? In other words, you take the experiments that you run to get differential activity in the brain, and you project those experiments into this similarity space. And then again, you see which processes are neurally real, or which processes need to be eliminated, which processes need to be split or merged, and maybe indeed what new task categories the brain recommends, what it treats as similar and what it treats as different. And again, I know you heard from Russ, and I'm sure he went over this, so I'm just going to essentially remind you of the kinds of things that we've been doing along these lines. So this is an experiment from Lenardowicz in 2010. They did a meta-analysis of hundreds of neuroimaging studies purporting to investigate the components of cognitive control. So the cognitive processes or the cognitive operations that are thought to characterize cognitive control include working memory, response selection, response inhibition, and task switching. And what they did was a discriminability analysis. So it's a k-nearest neighbor classification for those who care, but the basic idea is you're projecting these neural patterns into this reduced functional space, and you just see what clusters together. And you also then can do the opposite of that. What clusters together, and what can you actually tell apart from one another, right? So what we're looking to do is to see whether the neural activation patterns observed during the engagement of these subcomponent processes were different from one another and were indeed associated with, that is predicted by the construct in question. Here's just one piece of their findings. I'm not going to, again, I'm assuming this is largely familiar. So here's cognitive control, right? And the idea is we're comparing the neural activation patterns observed during these various tasks and seeing whether we can tell them apart. So here's task switching, here's response selection, here's response inhibition. This is cognitive control writ large. This bottom line is just bilingualism as a control condition. The point here is that task switching could not be discriminated from response selection, and task switching could not be discriminated from response inhibition. And so the notion is that if the brain doesn't show distinct patterns under these various conditions, maybe psychologically, these things are not in fact distinct. So there are many possible reasons for these results. There might be noise in the underlying label, so we're just, basically fitting garbage. It also could be that task switching as operationalized psychologically generally involves response selection and inhibition, so you wouldn't be able to tell them apart for that reason. It also could be that the successful discrimination here was founded on differences in paradigms used to isolate these things and not on the construct distinctions themselves. Nevertheless, it's also possible, and this is the possibility that this article floats for us, that task switching is a dubious psychological construct. That is, it's a candidate for elimination from a mature taxonomy of the mind. And why? Well, because the brain does not distinguish it from other constructs. And if the brain doesn't distinguish it from other constructs, the thinking goes, maybe we shouldn't distinguish it psychologically either. And again, the idea here is not that people don't switch tasks. The idea here is that all the various instances of switching tasks do not fall into a psychological or a psychological natural kind. There's no psychological reality to task switching. This is also Russ's work. I just want to point this out. Well, you'll see why in a sec. Here, the idea, the technique is a little bit different, but the basic idea is the same. So they took these 8 experimental conditions. They took the neural activation patterns observed in those 8 experimental conditions. They projected that into a 6-dimensional space. In this case, they just used a neural network with a 6-node hidden layer. And the idea here is the same. Can we tell the difference between these 8 things? And indeed you can. And this diagram here tells you how. So we have 6 dimensions. So risk-taking here, it's really far out in space along dimension 5, but it's kind of close in to the origin on the other dimensions. In contrast, classification is also far out in space on dimension 5, but it's also much further out in dimension 3 and dimension 2. That allows you to discriminate between those 2 constructs based on where in the brain the neural activations cluster, right? Again, this is like a 6-dimensional functional space. And similarly for the rest of these, right? You can see that it's pretty easy to tell them apart because they're all going to be in a different region of this 6-dimensional space. Now, I want to draw your attention to response inhibition here. And you'll see why in a second. Notice that it's very far out on basically all the dimensions except dimension 4, right? So it's in its own particular region of space. Now, what Poldrack and colleagues did is also do essentially a lexical analysis of these same data to see what kinds of concepts or words these dimensions load on. And so indeed, dimension 6 highly loads on response inhibition. It's the only dimension that highly loads on response inhibition. So dimension 6 seems to tell us something about response inhibition, right? But here's the interesting thing. Response inhibition does indeed highly load on dimension 6. Indeed, it's the only one that highly loads on dimension 6. So that echoes what we find. But look, it also loads on almost all of the other dimensions. So that tells us that response inhibition is response inhibition, but response inhibition is also a bunch of other stuff that we don't really understand. And that tells us we don't really understand response inhibition, or at least that we don't understand the way response inhibition is represented in the brain. And that might give us, again, some reason to rethink that particular construct psychologically. Because it's clearly not just response inhibition. It loads on, let me go back up, twice actually, right? So it loads on all these other dimensions, which include audition and working memory and speech and phonology and so on. And if that's what response inhibition looks like, that means response inhibition is actually probably not a primitive and is much more complicated than we have so far given it credit for. I've got a different gloss on this way of going about things. I think instead of using this to search for fundamental functions, I think what we should be doing with this kind of approach is to find the set of explanatory factors, this 6-dimensional space, that appear to have some psychological significance. I've called these neuroscientifically relevant psychological or NRP factors. Or if you want, by analogy, and I think this is maybe a useful analogy, what we're doing is not trying to find fundamental functions. Instead, we're trying to specify neural personalities, that they're different loadings on NRP factors. So is this brain region extroverted or introverted? Is this brain region sensation seeking or not, and so on. So what we do is we find these factors, and this is just a factor, this is actually non-negative matrix factorization. This is just a factor analysis of the same kind of data that Poldrack and others were using in the things I showed you a bit ago. And the notion is this dimensional reduction is sort of like inferring personality traits from observed behavior across circumstances. So what neuroimaging experiments are is we're observing the behavior of parts of the brain across different circumstances, just like we observe the behavior of individuals across different circumstances. Then we do dimensional reduction to extract some personality values. Same thing here. We do some reduction to extract some personality values. This tells us basically that these cognitive domains like speech, semantics, phonology, express functional features. In this case, they're all arrayed along the zeroth axis. And these brain regions respond to that particular functional feature that is present in those cognitive domains. And again, the way I've talked about this is that the brain regions don't have fundamental functions. They instead have functional dispositions. That is, they will have a disposition to respond to different circumstances. Anyway, the point here is simply that we have these ways of investigating existing data and using that existing data to change around the way we divide tasks. So we just can call this task reclassification. So what are the strengths of task reclassification? Well, the main strength of task reclassification is that it's data-driven, right? That is, it's rooted in large amounts of available data on human performance in controlled conditions. It doesn't, in that sense, rely on human intuitions about the nature of the task. So when we do a functional decomposition, we're largely relying on our intuitions about what that decomposition ought to look like. This ignores that and says, well, maybe, but let's just ask the brain what it thinks the functional decomposition might be, and what it's going to group together. And so we sort of bypass human intuition, at least to a certain degree. And it turns out, indeed, that it can yield counterintuitive or surprising results, as I showed you in the case of response inhibition, right? It's a much more complicated construct, according to the brain, than we've been treating it inside psychology. So this can be a very useful intuition pump. This is where human intuitions come back in, but now the intuitions are more constrained than they were in the case where we're simply doing a task analysis on our own. The other really important strength here is that this, unlike computational theory of mind, unlike cognitive psychology, this way of dividing up the psychological space explicitly includes brain activation patterns as part of the classification of psychological processes, right? So this is basically building the brain into psychology from the get-go. So those are some important strengths of this approach. What are the weaknesses? What are the negatives? Well, frankly, one of the negatives is that it's data-driven. In other words, it relies on the very same data collected under a potentially dubious paradigm, right? And the analytic techniques, including neural networks and whatnot, they don't typically offer unique solutions. That is, you can tweak parameters. You could have used a four-dimensional hidden layer in your neural network. You could, using different techniques, there are different parameters you can tweak. And so, in other words, you can maximize variance capture. You can maximize classification performance. You can maximize hyperplane margin width. You can depending on the technique you use, there are various things that you might be targeting to give you what you consider a good solution. And here's the trouble. Each of these things you can maximize don't have any quite clear relation to the thing we care about most, which is what is the accurate taxonomy to come out of this analysis, right? Which of these maximizations is the most relevant to that task? And there's simply no framework we have that will give us an answer to that question, which is not to say there isn't a framework we might develop that could help give us an answer to that question, but we do not have one currently. Now, and most importantly, in my mind, given that we're looking at experiments that are conducted in, say, a scanner under the guise of cognitive psychology, all these data are still based on largely ecologically invalid tasks and largely tasks that ignore evolutionary history. That, to me, is an extremely important weakness. And that, to me, also is why I was so excited to come across Paul's work, that tries at least to hit, approach the issue of taxonomy directly in dialogue with evolutionary history. And that brings us to Paul's part of this presentation, and I'm going to stop sharing now and let Paul do his thing.
Okay. Thanks, Mike. All right, let's see if I can do this quick. There we go. You can see my screen?
Yes.
You can hear my voice. All right. Well, thanks. So yes, I was also excited to find that people are asking these kinds of questions because this has also interested me as well. So I'm going to describe as Mike said, a different way of looking at it. That's, I think, complementary. Now, so the typical approach, as Mike noted, is functional decomposition, where we propose what are the distinct information processing functions in the brain, and then we search for the neural systems that implement them. And of course, the challenge is defining what those functions are. So what I'm going to propose is sort of a strategy for trying to do that from a different angle, looking essentially at evolution through a process that I've called phylogenetic refinement. But what it really is just applying the comparative method of biology to theorizing about the brain. And the logic is also very simple. The logic is that we all, I think, accept that the brain was produced by a process of evolution. Well, if we accept that, then it seems worth asking, well, what were the steps in that process? How did the brain get constructed over evolutionary time? And that allows us to, instead of defining the particular parts list based on concepts like cognition, attention, what have you, we can consider how the mechanisms and circuits and functions themselves were differentiated and elaborated over evolutionary time. Because each thing in the brain has an evolutionary history, and that provides strong constraints on how it could work, how it's organized, etc. And it turns out we have a lot of data about that history. There's a whole field of people who have been studying many different species to infer what is the history of our nervous system. And that, I think, provides an alternative and perhaps better way to ground a decomposition of the problem of behavior, to delineate the true natural kinds. Now, notice I'm talking about behavior here, not cognition so much. I'm asking a question that's a bit more basic about what are the pieces into which behavior is decomposed, and then cognition presumably emerges within that story. Now, before I go through that, I just want to correct some misconceptions that many people have about evolution. One is that evolution is nature's way to answering questions posed by the world. That's not really true, because unlike engineers or theorists, evolution doesn't even identify problems. It merely introduces variations into populations of animals and then favors those that happen to accomplish something that used to be a problem. Only after the solutions can you actually even say there was a problem that was solved. So there's no goal or metric that drives the process. And misconception two is that this yields optimal solutions. Now, that's not entirely true either. There is improvement in general and adaptation, of course, but they're far from optimal. And that's partially because for a variation to even enter into natural selection, it must first be possible. And the vast majority of potential mechanisms are not. They're simply not possible as variations of the ancestral state. And part of the reason for that is that the genome doesn't actually describe like a blueprint of the organism or a connectome for the brain. What the genome actually describes is a recipe for building an organism through a lengthy developmental process. And like any recipe, development is a sequential process. So each stage depends on certain assumptions about what happened in earlier stages. So you can't just modify an early stage, no matter how useful this might be, because it's going to violate the assumptions of later stages and produce a big mess and a non-viable organism. And so evolution is really left with only a few modifications that can be viable. And among those are elongating the developmental process, or differentiating system and then specializing them differently over time. In other words, the organizations that arise in descendant species are emerged through differentiation, specialization, and elaboration of earlier systems. And so it's massively constrained by the organization of that ancestral system. Now, I think this is actually really great news for us scientists, because this means that we can actually use evolution to help us carve up the problem of behavior, because this effectively gives us a kind of taxonomy, at least at the neuroanatomical level of neural organization. And so what I'm going to do is I'm going to take a kind of a brisk walk through evolutionary history, essentially to sort of summarize what happened along our lineage leading to humans. So here's the phylogenetic tree of animals expanded almost exclusively on the lineage that leads to humans. All these branches are gigantic as well, but I'm going to just follow along this one. These branching points are where we diverge from different species. And I'm really going to focus on very fundamental, very simple behavior. I do promise that in the end, when I pass it over to Michael, we're going to try to connect it to human cognition. And also I want to say, I'm going to follow through this incredibly quickly, and so maybe ridiculously quickly. It's going to be flashcards. And so it's going to be impossible really to assimilate much of what I say, but I just would like to draw your attention to the fact that the story is continuous, and that story is a continuous progressive differentiation, both at the level of structures and their functions, behavioral functions. All right, and I'm going to start at way back here somewhere, 3.8 billion years ago, approximately, at the very origins of life. And there's a general consensus that life began with autocatalytic sets. So these are self-sustaining chemical reactions, sort of in a circular organization. And that produces both replication and a basic type of metabolism. Now, metabolism is essentially taking some kind of compound that you need to maintain life and keeping it under control in a particular desirable state through a negative feedback control. Now, once you enclose these processes in membranes, you can now distinguish two kinds of metabolic control. A metabolic control that's entirely within the body, we can call that physiology. But there are some compounds that you can't produce internally. You need to absorb them through the world, and you may be in a place where there's not enough of them. Well, in that condition, you can just move, even randomly. And that's a type of control that extends to the environment. And for many years, it's been suggested that this is essentially behavior, that fundamentally behavior is about controlling your state by interacting with the environment. This idea has been around at least since Dewey, Piaget, and others. Essentially, that the task of behavior fundamentally is not really about building knowledge and all the kinds of things that we do today, but complementing the dynamics of the environment such that the organism-environment system flows towards desirable states, controlling your state in the world. Now, the first nervous systems emerged as a specialization of the ectoderm, the external layer of cells. And in these original eumetazoans, these neurons were distributed as a diffuse net across the cup-shaped body. And they specialized, there were two specializations: the apical nervous system, which was rich in chemosensory and photosensitive receptors, et cetera, and communicated mostly through hormonal secretions; and the blastoporal nervous system, which controlled contractions of the blastoporal, either expel water or suck it in. This part here will eventually become what we now call the hypothalamus, while this part here will become much of the rest of the nervous system. And so these animals foraged around the world through something called levee walks, where they made small movements, exploiting a local environment, and occasionally making a longer range movement, exploring to a new climb. And Thomas Hills and others have suggested that moderating between exploitation and exploration was the original role of the neurotransmitter dopamine. That tonic dopamine is telling you you're doing well, you should keep doing what you're doing. And when it drops, it's saying things are not as good, go somewhere else, see if you can find a richer region, essentially through secretions from this apical nervous system. This organization is still seen in cnidarians like jellyfish, but in our branch, the bilaterians, the body elongated, the blastopore fused into a tube that became the digestive tract, and the apical and blastoporal nervous systems merged at one end of the body, forming where the head will be and where the hypothalamus arises. This organization is still seen in protostomes, which includes all insects, annelids, cephalopods, mollusks, etc. In our branch, though, the body inverted dorsoventrally and the neural plate folded into a tube, creating the basic plan of all chordate nervous systems. All of our nervous systems are fundamentally a tube composed of these segments that enlarge and modify through evolution later. In the earliest chordates, this is sort of a side view of a hypothetical protochordate. There's essentially a hypothalamus projecting to a spinal cord. And this had this kind of, this is a top view, unfolded top view of that tube. You can see dopamine dependent search strategy. These animals also possessed an escape circuit with a photosensitive patch at the front of the head, a single patch that projected to a structure called the tectum that projected downstream. So if a shadow falls on the animal, it scurries away quickly. In early vertebrates, this single patch split into two that migrated to the sides of the head. And the initial bilateral projections became primarily contralateral. This is why you have the contralateral decussations. One explanation for it is because now, this was advantageous because if now a shadow falls on your right eye, you're going to first turn to the left before escaping. So you have oriented escape behavior. This then retinotectal system enlarged, conferring a kind of spatial selectivity. So now you have specific movements away from specific locations in space. And part of the tectum specialized in the rostral tectum with contralateral projections implementing approach behavior. Now you'll turn towards the site of stimulation. And the selection between approaching and avoiding something initially, at least, was very simple. Very simple key stimuli is the term from ethology, where, for example, if something is large and expanding quickly, you probably want to get away from it. These early chordates continued this kind of tonic dopamine-dependent foraging, but one important thing that happened is an expansion of part of the hypothalamus into what will become the telencephalon, composed of an external segment called the pallium, which receives sensory information about the animal's current state, and the subpallium, which will become the basal ganglia, which on the basis of that state selects things like approach versus avoidance. And this system could be trained by dopamine emitted in a much more phasic burst, saying that what you just did right now in this particular state was better than expected, and essentially implementing reinforcement learning, which was, of course, extremely important for our early ancestors. The pallium also specialized into two systems. One, the ventrolateral pallium, was primarily specialized for the exploitation of the local environment. So things like approaching, ingesting, learning what's good through associative learning, etc. And the second part, the medial pallium, specialized to going somewhere else, this other aspect of behavior, going somewhere else, navigating away. This part will ultimately become the hippocampus. Jawed vertebrates elaborated part of the hindbrain, essentially producing a system for canceling out sensory consequences, a system that canceled out sensory consequences through a number of mechanisms, including common mode rejection, reafference principle, etc., leading to the construction of what we now call the cerebellum, essentially a sensory motor structure that predicts the consequences of actions. And this was very important because it allowed our ancestors to overcome certain limitations in their lifestyle that allowed them to become bigger and faster and much better predators. So this was, of course, also very important. Bony fish further elaborated telencephalon, creating additional distinctions between septal nuclei, amygdalar nuclei, ventral stratum and pallidum, etc., still organized fundamentally around this idea of exploiting locally versus exploring somewhere else. Upon getting out on land, our ancestors encountered a dramatically different world with a larger visual range, with a wider variety of affordances, with novel demands like thirst, etc., and an overall leading to an overall expansion of interactive behavior that was accompanied by additional parcellation, particularly in the pallium. And different animals, different branches dealt with this differently. Amphibians, and this is a sort of a cross section through the telencephalon, the amphibians expanded more the medial pallium, this navigational system, perhaps because if you're an amphibian, it's very important that you don't lose your way back to water or you'll dry out and die. Sauropsids expanded. Sauropsids, which lead to the ancestors of reptiles and birds, they dominated the diurnal niche. So they continued to use and expand their tectal approach and avoidance systems, but also expanded the tecto-recipient parts of the pallium, the ventral and lateral pallia, which become the dorsal ventricular ridge, where a lot of the complexity of bird behavior, for example, arises. The synapsids, our ancestors, the synapsids, they became nocturnal. And so they actually lost trichromatic vision and rely more on olfaction, audition, etc. And they expanded this green part here, the dorsal pallium, into what we now call the neocortex, that became laminar with this inside-out development that allowed it to expand dramatically. So now we see more familiar structures emerging in this story. In early mammals, you get a parcellation of this dorsal pallium that becomes modality specific. You have visual region, auditory, gustatory, somatomotor, etc. And it's organized in all mammals as two sheets, according to Barbara Finlay and her colleagues. A dorsomedial sheet that's spatial, retains spatial topography. And so it implements sort of visual motor maps for different actions like walking versus biting, etc. Whereas the ventrolateral sector does not have the spatial topography, but identifies key stimulus features to evaluate and select which actions are the most appropriate at a given time. So this is already starting to look familiar, maybe to many of you. This is sort of like the dorsal and ventral streams of Milner and Goodale. In primates, primates returned to the diurnal niche and became very strongly visual and expanded the big sections of their neocortex, particularly the frontal, temporal, and parietal, so much so that the whole thing curled around the insula, producing a familiar shape of the primate brain. But it's still organized into these two sheets. A dorsomedial sheet that's spatial and contains a variety of action maps for the large behavioral repertoire of primates, and also going beyond basic key stimulus detection to object classification, recognition, etc. That's much more sophisticated, but still ultimately in the service of evaluating and selecting different kinds of actions. So now this is, I'm getting here to basic, relatively simple primate behavior. How do you go to cognition? How do you go to something like what humans can do, which involves a lot of mental calculation, etc. One possibility is that it doesn't, that there is really something fundamentally different about what human cognition is. But I think given the relatively continuous story we've had so far, it's likely that it continues to modify this organization to get us to more complicated functions. But I'm not going to explain chess playing. I'm going to discuss something a little bit more humble, a monkey sitting in a tree deciding between different goods. So he's faced with a berry here that's in reach, but he's also faced with these apples that perhaps much more appetizing that are out of reach. And so if here is his brain, these two goods and values may be represented in orbitofrontal regions, for example, and the apples are more appetizing. But the actions that are currently available, the affordances in the world, are things like there's a reachable berry and there's a walkable branch. The apples are not reachable. Now, if you can predict the consequences of reaching out to a berry, then you can say, well, that'll yield you this good. And that may be enough to bias your competition such that you select the reaching movement, get that berry. And that's fine. You'll survive, you'll feed yourself, and you'll do well. But you can, of course, also predict, if you're slightly more sophisticated, that you can walk out on if you walk out on the branch, then you'll bring the apple within reach. And so you can predict affordances that might arise on the basis of your actions. And then consequently predict that will yield you this more appetizing good. And maybe that will flip the competition here. So you'll choose to forego the small reward and walk out on the branch and get this good that you want. So it's of course far from chess playing or abstract thinking, but it's one step towards higher level planning, something Giovanni Pizzolo and I have called hierarchical affordance competition. What about language? Now, language is a tough one, and I'm not going to say too much about it because language is so much more advanced in humans that it's hard to imagine how one could explain it in this kind of framework. Nevertheless, if you look at a brain of a rhesus monkey or a chimpanzee or a human, they're actually quite similar in their apparent neuroanatomical organization, at least. One big difference is this arcuate fasciculus here shown in red, which is much more extensive in humans than even in chimpanzees. But can we think of language in the context of this control view of behavior that I've been describing? I think you can. I described control within the body, physiology, that's control within the body. Behavior is control through the environment, through interactions with the environment. Well, you can also achieve control of your state through other creatures. And imagine you're a helpless human baby and you can't obtain some substance yourself. Well, it so happens that in your niche, there exist these creatures called mom and dad, and they are willing to do almost anything to provide you with whatever it is that you need. If you just make an utterance, cry or make a gesture, they will come and they will provide that thing for you. And the wonderful thing about parents is that you can get very complex effects, extremely sophisticated behavior out of them through very simple cues. And more so, they will teach you over time which simple cues to utter to produce those complex effects, essentially scaffolding the behavior in that sense. Effectively, this is of course far from full-fledged language, but it's the fundamental aspect of communication, which is persuasion. And this talk is an example of trying to persuade you about something. Anyway, this has been a very brisk walk through evolutionary history, of course. You know, some of the conclusions that I think have been apparent along the way is that the story has throughout been really not about building knowledge, but about controlling interaction with the world. Those are the fundamental distinctions that emerged in this process. Now, that's not to say that there isn't knowledge in there, but it's not the most fundamental aspect. And that the story of the evolution of the brain and its behavior has been this continuous expansion of that control. So this leads to a different conceptual taxonomy. It doesn't necessarily give us these kinds of conceptual categories or ontology, but rather this. And this taxonomy here actually is kind of a summary of the talk I just gave, of how the different aspects and different regions diverged from each other over actual evolutionary time. And the advantage of this taxonomy is that it actually does map quite well to the brain, even to specific subregions of the cerebral cortex. I think in a way that's promising. So anyway, if you want more detail on this, that's described in this paper published last year, as well as a paper with some colleagues where we used this kind of approach to deconstruct the question of what attention is. Now, I'll just return to this briefly, the strengths and weaknesses of this approach. The strengths are that it respects biological reality. The brain really did evolve through a sequence of elaborations. And along the way, each step really did constrain the next one. And so for that reason, to ultimately understand the brain, a continuous story is necessary. And some of the results of that process are just not going to make sense otherwise. It's going to be impossible to make sense of certain things, except in the context of this history. And the one question I would give you is, why is the human retina inverted? Why are the receptors facing the wrong way? And I think the only way you'll get that answer is to look at evolutionary history. If anybody's interested, I'll go through that. And furthermore, there is growing data allowing us to infer that sequence. And the data is based on techniques that are really advancing rapidly. And they provide really a very independent source of constraints for theory building, very independent from the kind of imaging or behavioral studies that Michael and others have shown. And I think it avoids the trap of prematurely defining or labeling the concepts. The kind of typological thinking that we tend to engage in as scientists can quickly get trapped in false concepts if you don't define them correctly. I don't think I have to make this point with this audience. This method allows what Ernst Mayr called population thinking, where you can think about things emerging out of a distribution of related functions that's more or less continuous. So you can express the gradual emergence of ontological distinctions, because every distinction that does exist in biology actually does have some kind of history of emerging out of something where it wasn't a distinction. And we can, I think, empirically define that history. However, the approach has some weaknesses. And I think the biggest one for us humans is that any comparative approach is limited by the surviving species. And so we really can't use this method to cover that last 6 million years of human evolution. And that has been a very interesting 6 million years. A lot has happened. Chimps have very sophisticated social structure, tool use, communication skills, etc., but really still very far from the human level. So how to fill that gap? Well, we can use fossil data, but that just lets us infer about lifestyle, etc., and maybe some capacity, but not about the actual structures underlying those. Developmental data may tell us something, but it's really ethically impossible to access it in humans, and in any way, it's potentially misleading, because if you don't have multiple species, it's hard to compare, to infer evolution from development. So the end result is not really a theory of human behavior. However, it's really just a theory of primate behavior. However, I would say that that should still be useful, even for psychologists, because it provides a set of baseline constraints for theories of human behavior. Whatever happened in the last 6 million years still has to be compatible with that, or with those origins. And so it provides a kind of a potential grounding for data-driven approaches like the ones that Mike described. So with that, I'm going to pass it back to you, Michael. And I'll stop my screen share so you can do yours.
Great, thank you. And yeah, so this is a slide I've shown already, but just to remind you. So here are some strengths of task reclassification, that it's data-driven, that it's rooted in these large amounts of available data on human performance, and it does indeed attempt to explicitly include brain activation patterns in the actual classification of psychological processes, something that we would like to be able to do. And then the weaknesses again, right? That the same data have their own weaknesses, and any analysis based on them is going to inherit those weaknesses potentially. But the thought here is that the phylogenetic refinement and task reclassification may have complementary strengths and weaknesses. And how might that go? Well, so here's just a series of questions that Paul and I have, and that we like to share with you. So indeed, can our knowledge of the chimpanzee neural system and behavior repertoire constrain a hypothesis space for task reclassification, right? We're using this task reclassification as an intuition pump, but we also want to make sure our intuitions are tracking ecologically or evolutionarily valid categories. The analysis that Paul provides may well help us do that. And even if we can't do this directly, can phylogenetic refinement drive hypothesis generation for new experiments? And I'll show you an example of that in a little bit. And in general, a question that we have that we think is worth exploring is can the constructs discovered by task reclassification, such as the NRP factors that I mentioned earlier, can those constructs face both ways? That is, can they be used to bridge human neuroscientific data and evolutionary or developmental data? Again, generally, is it going to be possible to map the task categories? And this is just the converse, really, of the question I asked a second ago. Is it going to be possible to map the task categories revealed by phylogenetic refinement to their human analogs? And if we can do this, can the human analogs of approach, avoid, interact, explore, exploit, can these help us better understand primate social behavior or foraging decisions, for instance? So in other words, the idea is that there may be a two-way interaction here. We better understand primate behavior because we have the human example. Then better understanding primate behavior helps us better understand human behavior, and so on, in what we would like to think might be a virtuous cycle. And then when a system is exapted, right, and I'm going to show you again his slide about the particular brain systems that were differentiated over evolutionary time and what their basic function is. When a system like that is exapted or reused, a question is how much of its original character does it in fact retain? And then how much insight is therefore possible when we establish that such an exaptation has happened? So if the approach avoid circuit is in fact been exacted in humans for social behavior of particular kinds, what can that tell us about social behavior, given we know something important about the underlying circuit and its evolutionary history? So this is the slide he showed you. Let me, yeah, there. About the different taxonomy. So here's the familiar taxonomy, the familiar task analysis we get from cognitive psychology. Here is an alternative that we get from phylogenetic refinement. And I'm just going to go up into the, the lower regions here, right? So we've got exploit, explore, we've got interact, we've got approach and escape, we've got, locomote or ingest, and then we've got these various particular action maps. So one question is, can we use these sorts of categories to reinterpret the kinds of experiments we've done in psychology, and equally importantly, or perhaps even more importantly, can we use these kinds of categories to suggest new experiments or ways to interpret experimental data that we collect? So I want to tell you just briefly about some work that's going on at University of Western Ontario right now. This is work coming out of Jody Cullum's lab, one of my collaborators locally. And one of the things we're trying to do, and you can imagine it's quite difficult to do this properly, but we're trying to make experiments done in fMRI scanners a bit more ecologically valid. So we're using virtual reality. In this particular case, what we're trying to do is actually have people play Pac-Man in the scanner. Why? Well, for one thing, that means people are actually acting, or at least virtually acting. It's better than just lying there and looking at stimuli. But also think about this for a second. What is Pac-Man? Well, it's got a bunch of ingestion that's occurring. It's certainly got approach avoid. You want to eat the fruits that appear, and you want to eat the dots as they go, and you want to avoid those ghosts. So we could analyze this in terms of classic action selection paradigms, from cognitive psychology, but it seems we can also potentially interpret this in terms of the categories that Paul is revealing to us from his analysis of our phylogenetic history, and not just those categories, but where in the brain we think the action is. So when we play Pac-Man, will we see ingestion activity? Will we see approach versus avoid activity? Will those differential things show up in the brain as we behave in a slightly more naturalistic way inside a scanner? We don't know the answer to this, right? Again, we mentioned this already earlier. I mean, in COVID times, not a lot of human experimentation is going on, but this is the direction we want to go. And the kinds of analyses that Paul is offering may offer an avenue to a way of interpreting these data in terms of differential brain activation that gets us closer to our phylogenetic history. And I think if I remember from our discussion earlier, Paul, you also had something you wanted to bring up along these lines.
Yeah, if you could bring up the conceptual taxonomy slide, I can do it.
I just did. It's not.
Oh, yeah. Okay. Hold on. I gotta bring myself back. Yeah. So it's actually there's two examples that I could say. One is the an idea I mentioned about the early role of dopamine way back at the root of the animal phylogeny, between exploring and exploiting, sort of sticking locally versus exploring somewhere else. And the people, Thomas Hills and his colleagues who have phrased this hypothesis have also suggested they've taken it all the way to human cognition as well, basically focused strategies in cognition versus diffuse, et cetera, and the different kinds of pathologies one gets with different kinds of dopamine disorders, et cetera. So at least they've suggested that this could be connected. And the other paper there that I cite by Hamel et al., what we've done there is we've looked at the question of attention, which has for a long time implicated the parietal cortex, but in very confusing ways that don't match to the normal sort of criteria for something being attention versus intention versus something else. In that paper, we describe how it may be an aspect of orienting behavior, which does involve regions of the parietal cortex. It's the action map for orienting behavior related to superior colliculus, et cetera, leading to spatial attention, as you can see there in the taxonomy. So we've attempted to do this for some concepts like attention. I try to do this for things like decision making, but I think in general, it is possible, at least in principle, to connect these concepts to specific regions of within more complex cognitive tasks. At least that's my hope, but we'll see how that pans out.
Yeah, great. And now you also might have some skeptical questions. You know, if this is the kind of taxonomy that evolutionary history can give us, then we might say, that's not good enough. And so to what extent should psychology be constrained by the identification of these basic functions of evolutionary preserved neural systems? Maybe that's just not differentiated enough to be useful for human psychology. And importantly, although you know, Paul touched on how you might think about language within this framework, we have to ask the question, how big a barrier is language, right? Did language in fact cause a significant enough cognitive reorganization that it threatens the continuity thesis for cognition? And that is what we have for you today. And appreciate you listening and looking forward to hearing your questions and comments.
All right, thank you very much, Paul and Mike. One of the downsides of Zoom webinar is that we cannot clap. But I'm sure all the attendees are there. Let me remind the attendees how we're going to be proceeding for the Q&A. If you go to the bottom of your screen, you can see a Q&A button. Please click on the button if you have a question and write your name in the text box there, and I will promote you to the status of panelists. And you can then ask a question directly to Paul and Mike. I see that someone has a question because Rick, I'm going to promote you to the status of panelist. All right. Good. All right, Rick, the floor is yours. Rick, please don't forget to unmute yourself.
Exactly. Hi, thank you very much, both of you. Excellent talk and very thought-provoking. The question that occurred to me is really a clarification question, but it does fit in very much with what Mike was talking about at the end in terms of these skeptical questions. Because I was thinking a lot about cultural evolution and exaptation, and what reason would there be to suppose that the low-level taxonomy for, you know, that evolutionary history shows us is not going to be obliterated by you know, sort of very strong cultural factors that are going to come in, for example, after the appearance of language. I mean, as far as I can see, we're going to get just as complex types of organization between those basic categories that as we're getting with, you know, the earlier stuff, Mike, that you were talking about in terms of you know, function mappings. So do we have any reason to be not to be skeptical but to be optimistic here?
Yeah, I mean, I think so. So you know, I'm a big fan of Morten Christensen's work on language, if you're familiar with that. And his basic line is convincing to me, which is, look, language can't have been as special as some people think it was. Because if it were that weirdly special, our brains could never have assimilated it, right? So language itself had to evolve or develop, if you want to prefer that word. It had to develop so to fit the brain that was capable of taking advantage of it. And so the notion that it induced a massive reorganization in that case looks somewhat less plausible. I mean, I understand some of the reasons for thinking so, but the idea is here that no, like with any other artifact, that artifact has to have a two-way fit. It has to fit the world and do the things we need it to do, but it also has to fit the brain and the control system of the organism that's utilizing it. And so if you think of language as a very sophisticated artifact, then it looks like you can potentially explain it all in the same kinds of terms. And that's the sort of thing I try to do in the language chapter in my book. And I think it's the kind of thing that Paul was gesturing at briefly in that part of his talk where he touched on language.
Yeah, and I would add that, you know, if we believe that language is fundamentally a social, a tool for social interaction, then we can at least get a very good basis of social interaction by studying primates and still applying this comparative technique. So if we had a good theory of primate social behavior that's strong enough to satisfy us about chimps, we'd have a pretty good basis to then make theories about language that are better than if we didn't have that basis. Nevertheless, though, I think it is a distinct possibility that a lot of things will come in upon that basis that are not necessarily foreshadowed in all the things that I described. And I think it's almost like saying it would be helpful, but we shouldn't expect that will give us the whole story. That if we had a model of a chimp brain, which is but a long way off, of course, but if we had one, we'd be better off. But we shouldn't expect that it's going to get us all the way there. I mean, there's going to be cultural evolution, if anything, and our cultural evolution is much more sophisticated than any animals, I would say, obviously.
Right, but just again to reiterate, the notion is that there isn't necessarily an unbridgeable gap here.
Yeah.
Thank you very much.
Thank you very much. Olesia, the floor is yours.
Hi, thank you very much for your talk. My question is for Mike. So you have talked about various reasons for why we might want to reform the cognitive taxonomies that we have. Could you also comment on the reasons for why we might possibly want to keep the old categories, even though they don't correspond to what the brain recommends, as you have said? So when you were talking about response inhibition versus task switching, I was wondering if the category of task switching if we could say that it has richer descriptive content, that it could be more useful for certain pragmatic aims that we have in science, et cetera, et cetera.
No, absolutely. Look, it depends what you want out of your psychology, right? And you know, this particular, it's not a movement exactly, but this particular line of thought has come about because people have wanted now to tie psychology closer to neuroscience than has traditionally been the case. And when you try to do that, at least if you try to do that the way that cognitive neurosciences have done it since the 70s or so, you find that it's just a big freaking mess. Now, I don't think it's actually a big mess. I mean, I've got a story as to why we see the kind of patterns that we see. But for a more traditionally minded person, it just looks like a big mess. But if what you care about is to get some kind of handle on human, say, performance in various kinds of tasks, like memory tasks, like action selection tasks, like cognitive control tasks. If you want a kind of explanatory framework, you know, that features representations, that features transformations of those representations, that can give you an explanation of a mechanistic explanation in computational terms as to why people perform this way rather than that way under those circumstances. Cognitive psychology is a perfectly adequate science, right? It's got a great explanatory power. And so, but for me, that it's instrumental, right? That gives you a handle, and then, you know, and you can even do things like predict what kinds of workplaces will be more productive for people, given what we know about attention, for instance. And we can use it to predict what kinds of situations in which people will remember more things than others, and which things they'll remember. So you can engineer your environments using these insights. And so if that's the sort of thing you want out of your psychology, then there's really not a lot of motivation for mucking around with what we're up to. But as I say, if you're interested in tying it closely to the brain, right now, those mappings look quite complex. And that gives us a motivation for saying, okay, well, if we really want, you know, what kind of taxonomy do we want for cognitive neuroscience versus a kind of autonomous cognitive psychology?
Thank you.
Paul, do you want to add something, or?
Oh, well, no, I guess I could echo a lot of what Mike said, but I could add one thing, which is in the usefulness of a different taxonomy. In decision making, which is really what I study in my lab, there's a very big debate, as many of you probably know, about, for example, whether the basal ganglia are involved in decision making. And there's a large camp with beautiful data saying yes, and a large camp with beautiful data saying no. But when you look at the at least in my eyes, when you look at the evolutionary story of it, you can see that there's different kinds of decision making. There's the choice between which aspect of your behavioral repertoire you want to use, which is a basal ganglia, which is, at least from this perspective, assigned to the basal ganglia. And then if you've chosen to do a particular type of behavior, then the decision that you make within it is a more cortically based decision. And our studies and others have suggested that, maybe that's why when you do experiments with people doing something like choosing to go here or there, it doesn't look like basal ganglia is involved, because basal ganglia is chosen, I'm going to do one of these reaching selection tasks. And that's the hierarchical level at which it's making a decision. And so I think that all makes good sense in this framework. And I think it leads to some experiments that one might want to do to explore it a little bit more. So there is a reason, there's a motivation there. But again, it's what Mike said. It's what, if you're interested in how these things, like something like decision making, maps onto the brain, then it's a useful perspective. I'm not sure it will help, but it could also be helpful in the clinic, because there will be certain things that will be relevant to dopamine deficiencies and things like that.
Adina, I believe you had a question.
Thanks. I feel like I have many questions, but I think I'll start out by, I think Mike identified this sort of the problem that we're possibly radically wrong about what the fundamental functions are of cognition. And although I thought this was a fascinating suggestion, when I think of it in that context, I wonder how far it'll get us. So I appreciate that we're seeing functions that are simple enough at the early evolutionary stages that we can sort of identify them unproblematically. And that might give us some kind of foundation for some aspects of some sorts of tasks that humans do. But I guess I'm wondering how far that'll get us. I mean, those kinds of taxonomical distinctions are still very dependent on our sort of, our conceptual scheme to begin with. And then if Mike is right and we've got neural reuse, how much leverage does the original behavioral task get us in identifying what the reuse is for? So yeah, I'm just trying to wrap my head around the suggestion, because it's it's a fascinating perspective, but I'm not sure it's not subject to the same kinds of worries that that our original questioning of our ontologies was subject to.
Well, I can start with that. I think knowing what knowing what the parts list that we inherited is would help us know, at least conceive of the plausible acceptations versus the implausible ones. You can imagine how, for example, a human could use their orientation system to sequence their actions around the world into some kind of structure, and thereby solve a problem that's relatively complex and abstract with mechanisms that were originally adapted to less complex functions. And maybe that will therefore be a sort of a more evolutionarily plausible strategy of reuse. How did we use the pieces that we had to do the things that we do is kind of dependent on knowing what those pieces are. And I think that's kind of the way it would, I would be very happy if things turned out in a way where you could actually make that connection and say, well, so, one potential mechanism, make a theory about potential mechanisms using the inherited parts list, and then test that theory. You could design experiments to test between two specific theories on how some cognitive strategy evolves based on different combinations of the pieces we inherited. you know, that's maybe wishful thinking, but I think it's worth trying, worth putting some work into. I don't know if you wanted to add something like.
Well, no, I think it's exactly right. I mean, I don't know if you're familiar with the work I did with Barb Finley a few years ago, but that's exactly the story we tell, that reuse is not a problem here so much as an opportunity in thinking about, if we start with these very basic building blocks, how do we get really complex human cognition out of it? Well, reuse helps us understand how that's possible, because you can essentially layer recombinations on the basic building block, list of basic building blocks. And it's really important we have those basic building blocks, because of course we know that indeed we all achieve species typical behavior and species typical capacities. And if it were just a free-for-all, that would be an absolute miracle. And so the notion is, if we start with these building blocks and then we start with a normal developmental trajectory, then it's the combination of the, of the circuits in various ways. We find the right combination of things to do the more complex thing, the new behavioral task. And then, you probably don't know about this work because I've only published a couple of things on it, but I'm really trying to think hard now, not so much about reuse, but about constraint and the role that constraint plays here. Because the idea is that, these individual circuits are basic, but they're also really promiscuous, right? Approach avoid is all kinds of things, right? And so how is it that you constrain approach avoid to do a very particular thing in a particular circumstance? And here again, the notion I'm trying to develop, at least conceptually, and also empirically, we're doing some work on this if we ever can get back in the lab, is part of what happens in the interaction between brain regions is there's mutual constraint that comes into play there. And so some subset of the capacities of these basic circuits get selected and come together. And so it's not that you're employing the entire approach avoid circuit or something like this. You're constraining its activity in a particular way and combining it with another constrained circuit. And those two things together give you some kind of much more controlled or much more nuanced behavioral repertoire than you might imagine you could get just out of saying, well, it's approach avoid, it's ingest, it's interact, and whatever. Those are much too general to account for human cognition. So I'm again trying to develop this story about the importance of mutual constraint between interacting brain regions that could allow for a more fine-grained behavioral repertoire than you'd get simply by imagining the activation of those circuits. But I'm a long way from having a complete answer there. You once joked about my book that was everything Anderson knows or thinks he knows about the brain. It's pretty clear you did. That's what you said. It's pretty clear the next book is going to be everything Anderson doesn't know about the brain. And how constraints work is going to be an important part of that story.
So can I follow up for 1 sec, Edward, or do you have a list?
Go ahead, we have 2 more people and we are 53, so a short follow-up.
Okay, I think that makes a lot of sense to me if we actually are looking at mechanisms as opposed to sort of general functions.
Yeah.
And it seems to me that even at the level of delineating the big evolutionary changes between phylogenetic groups, we're not at the level of mechanism for the most part. We're still at the level of sort of big generalized functions, and then it looks a little bit harder to me to run that story. But anyway,
there's a couple of cases. There's a couple of cases where the mechanisms are fairly well pinned down within the context of the evolutionary story. The cerebellum is a good one, and some of the approach avoidance circuits. These are very well worked out by some groups, like Grillner's group, and so certain aspects of it are actually very well worked out. Steve Wise, Betsy Murray, and others are working on a lot of the primate frontal cortex, Dick Passingham, as well as others. So there are certain regions where it's being worked out in a lot of detail, in a satisfying level of mechanistic detail, and other places where, please come in and figure it out for us. Was what I would say. We need people studying all those transitions. Before it's really complete. But I do think there's some cases where it's actually at the level of mechanism, computational models, every level you could want, circuits in very, very good detail. That's worked out quite well. Others, no. And I think some of the most interesting ones is where it's not worked out.
Thanks.
All right. Linus, the floor is yours.
Hi, thanks Mike and Paul for the wonderful talk. So my question would be in a way following up on Adina's suggestion. So on the one hand, we have all this neural circuit that's evolved to do specific tasks in our phylogenetic history. But on the other hand, we have this modern task. They may be really different from those tasks that we evolved to do. So my question is, just want to hear you guys talk a bit more about how learning and development could actually bridge the gap by maybe recombining those phylogenetically old circuit efficiently to accomplish modern tasks. So one possibility is that we might actually have we have evolved learning or development mechanism for doing this, right? So we are particularly good at recombining those old circuit to serve new functions. So I'm just wondering what your thought about this.
Well, one is the neocortex does have a particular structure, and it does, at least according to many theories, provide a kind of a resource that could be used to learn very novel things in very novel ways. And the plasticity of the neocortex is very impressive. And so there's at least the potential that once you get such a structure, it becomes sort of generalist enough to be able to learn rather than evolve particular mechanisms that then solve tasks in very different ways, even maybe to the level of classical ideas about virtual machines. So I think there's in principle no reason why that can't be the case. I think the data sort of tempers that a little bit when you consider how different our life is compared to the life of a monkey. And yet, when we do experiments, similar experiments with humans and similar experiments with monkeys, we actually do have similar types of activity patterns, similar circuits involved. So it's not a complete tabula rasa that can learn everything. That's clear. But it is also a pretty powerful learning circuit, and perhaps it does allow us to take a Cro-Magnon brain and do quantum physics. But I think actually if I were to bet on a future insight into this, I think it will probably have to more do with cultural evolution. That it's because we offload so much of our skills into the society around us, and we're sort of living, we're sort of I forget the term, scaffolded. We have a scaffolding that allows us to do all these things based on interaction and sort of internalizing the logic of language and things like that. I think maybe it's there, but I don't know. I don't actually study those mechanisms. I think other people probably in the audience have much more to say about that, and Mike probably as well.
Well, I'll just mention, and I'm going to mention the book again, only because I know that it's escaped, and you can get it for free online if you're willing to go to those sorts of sites. But I do have a world example of how existing circuitry could have been co-opted for mathematics.
Yeah.
So how do you learn mathematics? Well, you treat mathematical equations as diagrams, and you interact with the parts in a very analogous way that you interact with physical objects. So basic capacity for tool use is actually what gets co-opted into our capacity to learn mathematics. And it's a really fascinating story. It's not my data. I'm just kind of reporting it in the context of trying to understand exactly the thing you're asking about.
Yeah, it's a good example, actually, of again, if we knew the pieces, we could figure out the strategy that's being used for these new cognitive tasks.
All right, thank you very much. When you'll have the last question.
Oh, I get the last one. Oh gosh, it's so much
Very
pressure.
good,
God,
yeah.
why did you have to say that? God. Hi, Mike and Paul. Thanks very much. Paul, it's, I guess more for you, although it's about maybe whether attention provides a kind of different test case about the classical view, Mike. So this might bring you into. So Paul, you mentioned your paper about no one knows what attention is. And I get that a lot. What I'm struck by is that James said something a long time ago, and in looking at the results of the phylogenetic approach in that paper of yours, you seem to converge on the same thing. And I take it that William James was expressing something that was sort of folk psychological. So roughly he said, just to remind everyone, attention is the mind's taking possession of one among many possible things or trains of thought. It involves withdrawal from, so that came up earlier in some experience, it involves withdrawal from some of the things to deal effectively with others. And there, I think it's pretty clear the conception is that it's fundamental to being able to act appropriately in the world, which is, I think, the picture you come up with more or less when you look at the phylogenetic approach in that paper. So I'm struck that there's a convergence, which what I would take to be a kind of folk psychological conception in some sense that James articulates, and the phylogenetic approach. And so here's the question. Might there be some ways in which the old classical approach does get it right, and we just have to figure out exactly how to do it? I mean, it might be that if that attention is somehow special in this way, that there's a way in that we have special insight to it that we wouldn't have for working memory or some other aspects. But the convergence here between those two positions struck me, and whether or not it speaks to something like, let all different approaches take their shot at this. Sometimes, one will work and others will as well. So
I think what we converge to in that is that there's a certain kind of attention that has sort of its roots in the orienting circuits of the rostral tectum, which is the superior colliculus, which is involved in orienting in primates, and the parietal cortex as well. And one of the take-home messages there is interpreting the parietal cortex in terms of the concept of attention is confusing. It doesn't have some of the things we'd expect, and has a bunch of other things that we wouldn't expect. And hence the intention-attention debate that's been going on for now 3 generations or 4 generations of neuroscientists, where the same region seems to be involved in attention, or which is sort of thought as the input to cognition, and intention to act, which is sort of like the output. And yet what we're presenting there is has been said before, that the parietal cortex, what it's really doing is controlling your gaze. And this particular part of the parietal cortex and superior colliculus are controlling your gaze. But because we are visual animals, so much of gaze essentially has such a strong influence on everything else we do with our bodies. What we reach for is constrained by gaze. Where we walk is constrained by gaze, that it takes on a kind of an executive role. This system, which is fundamentally, from an evolutionary perspective, a gaze orientation kind of system, originally for approach, becomes reused to organize the rest of your behavior once you are a visually driven primate with the fovea. And it just takes on that role because so many of the other systems depend on where you're looking. And so it is a convergence with some of James's view, because it is, but it's not, it's completely unrelated to other kinds of attention, like feature binding and other aspects of attention that have been seen. So it doesn't carve up the same way. It's much closer to action selection, really, than to any kind of input to cognitive systems. Now, it's essentially saying that, it's not going to be easy to map one version of attention to the other version of attention. But, we at least claim in that paper that the version that we propose maps better to the brain. But it does not address many interesting things about attention. It has nothing to do with them, presumably. And it's kind of the same story that I focus on in studying decision making. It's not one thing. There's lots and lots of different kinds of decision making with different circuits, et cetera. So, I mean, I guess that's my answer to attention specifically. Now, for other functions, you know, I think actually the phylogenetic approach will lead to exactly the same conclusions that other approaches have led. Reinforcement learning, forward models in the cerebellum, all these kinds of things fall right out. They're very natural to this phylogenetic approach. But the other thing I would add, I think, which I think Mike would like, is that embodied cognition and ecological psychology come right out of this as well. They're a natural fit to a lot of these ideas. So some people won't be so happy about that, but maybe.
Great.
All right. Except if Mike wants to add a sentence.
I'm happy to stick with it all leads to ecological psychology. That's a good place.
That's a good conclusion. Great. It's been really good to have the two of you with us today. Thanks again for this wonderful talk and who's bringing new ideas to the debate. We will see hopefully many of you next week, Dina and I, for the next meeting of the Cognitive Ontology Seminar. And on this, except if Dina wants to add something. Okay, you're mute, but I thought that was a no.
That was a no, but thank you everybody for coming, and thanks to Mike and Paul.
Yep, thanks for the invitation. Hopefully we'll get to see each other in person at some point in the
I hope so. We hope to be back at the center next year, sometimes during next year, and to reorganize real events. So when you bring more people back to the Center of Philosophy of Science next year in person. So hopefully soon in the future. Bye guys, and thanks again. See you next week.
Bye.